Hi, my name is Jacob Rook, and today we're going to talk about G protein coupled receptors. So, this is the basic model we're going to work with. We have a G protein coupled receptor in the shades of green. The region inside this red line is the outside of the cell or anything extracellular. The region inside the baby blue line is the phospholipid bilayer or cell membrane. And the region inside the yellow line is inside the cell or anything intracellular. So now I'm going to introduce some terms that will be associated with the rest of the video. This is the G protein coupled receptor, where the receptor is indicated by the arrow. The subunits of the G protein, where beta and gamma subunits are in the cell membrane, or the yellow boxes, and the alpha subunit is connected to the beta and gamma below, as indicated by the blue triangle. This structure is referred to as a heterotetrameric, which is being a macromolecule composed of three subunits of which at least one differs from the other two. Also notice that the GDP, or guanosine diphosphate, is bound to the alpha subunit. This will be important to remember for the rest of the video. The ligand, or a soluble molecule such as a hormone or neurotransmitter, is the specific molecule that was released that will activate the G protein coupled receptor. Notice that the black triangle shape is specific to the receptor. This is an example of a metabotropic receptor, or ligand-gated channel. This receptor is a type of membrane receptor of eukaryotic cells that acts through secondary messengers. Now let's see how this all works. In the first step, the ligand binds the receptor site and activates GPCR, or G protein coupled receptor, as I'll refer to it. This causes a conformational change in the receptor protein. The result is that the conformational change will ultimately affect the G protein, thus it is called the G protein coupled receptor. After the conformational change, the GPCR activates the G protein by activating the alpha subunit, which is mediated by GEF, or guanine nucleotide exchange factor. This GEF transduces the extracellular signal from the neurotransmitter from the G protein coupled receptor to the G protein. The end result is a removal of the alpha subunit, and now GTP, or guanosine triphosphate, is bound to the alpha subunit. Now that the alpha subunit is activated, it seeks adenylyl cyclase. Adenylyl cyclase is an intramembranous complex that is responsible for converting ATP, or adenosine triphosphate, to cyclic AMP, or as I'll refer to it, CAMP. The CAMP will go on as a messenger for cellular events, and the beta and gamma subunits will stay put in the membrane. We will come back to them a little later. Now we're going to take a closer look at the messenger action of cyclic AMP, which has numerous effects. First, cyclic AMP activates protein kinase A, which is also known as CAMP-dependent protein kinase. This molecule serves several functions that we will address in just a moment. Second, cyclic AMP can also be converted to 5' AMP, which is mediated by phosphodiesterase. Phosphodiesterase degrades the phosphodiester bond in the cyclic AMP. They serve to regulate the localization, duration, and amplitude of cyclic AMP, as well as other molecules. Protein kinase A is then mediated by phosphorylation, or the addition of a phosphate group to a protein or other organic molecules, to cause a cellular response, including mostly cellular metabolism, but also gene expression, cellular secretion, and affecting membrane permeability. The last step in this cascade is one of signaling to the next is the 5' AMP inactivates the protein kinase and thus regulates the system further. Lastly, the alpha subunit needs to be dealt with. The process of hydrolysis returns the alpha subunit to its original state, where it is now bound to GDP instead of GTP. This concludes the video on G protein coupled receptors. Thank you.